as we discussed a moment ago, we know that glucose is going to be reabsorbed by facilitated diffusion. So there is going to be a point that these facilitating transport proteins are going to potentially become saturated. If this happens, then we will see that glucose will then exit into the urine. So this is how this can be one potential sign of diabetes. If the glucose levels in the blood are exorbitant because the glucose is not entering the cells, which of course is a symptom of diabetes, then the glucose is going to stay in the blood. The blood will then be filtered through that glomerulus, and now typically what we see is reabsorption of glucose. However, if those facilitated transport proteins become saturated, we will then see an increased amount of glucose in the urine. Now, that is called glycosuria, which is going to be one indicative symptom of diabetes. Now, these can actually change by, which we'll talk here shortly, but just as an FYI, these facilitated transport proteins that are going to aid in the amount of allowing glucose to be reabsorbed can actually be changed genetically. So they can, the uptake of the production of these transport proteins can increase, and there can be a good amount or more being produced to where it will help with that facilitated diffusion. But this is indeed why we will see this glycosuria or glucose in the blood for symptoms of diabetes. Now, as we look at this tubular secretion, so we've had tubular reabsorption in the proximal convoluted tubule, and now we're going to see that some things are going to be then secreted back into the nephron. So some of that urea that was reabsorbed is going to end up back in the nephron, as well as some drugs. Again, this is why drugs are cleared. Some drugs are cleared with the kidneys because they will then be extracted. So tubular secretion means that things that have been reabsorbed, let's say, if you recall, this nephron is laying in that renal pelvic area. As we are reabsorbing things into the tissue, we are now wanting to secrete back into the nephron things that we don't necessarily want. So again, those nitrogenous wastes, as well as some of those drugs that we will see. Now, as we extend down from the proximal convoluted tubule into that loop of Henle, or the nephron loop, we're going to see that it has thick segments and thin segments. So in the thick segment, we're going to see that the reabsorption of sodium, potassium, and chloride is going to be about 15%. And then we're going to see that it's going to reabsorb very, very little water here, whereas the thin segment is going to be the opposite. We're going to see a process by which is called the counter current multiplier that's occurring within the loop of Henle. So we're going to see that as it extends down into the thin segment, we're going to see that the water is going to be reabsorbed. So it's going to be permeable to water on the thin segment. And on the thick segment, it's going to be permeable to those solutes that we were just talking about. And how they are going to be reabsorbed is the fact that it has proteins that these solutes are going to bind to. So we're going to see for every sodium molecule, we'll see 1 potassium and 2 chloride molecules passing as well. Okay, so as we further on, we'll talk a little bit more about how this process works when we get to that countercurrent multiplier. But it is important to know that we are going to be seeing reabsorption of water. So water is then going to be extending out into the tissue, while on the thick segment, we're going to see solutes are going to extend into the tissue. And that's going to be important for as water conservation comes into play when we talk about it here shortly. Now, furthering on from the loop of Henle is that distal convoluted tubule, which now is going to be controlled by those hormones that we were talking about before. If you remember, the aldosterone, the antidiuretic hormone, and the parathyroid hormone is going to be necessary for calcium. So we've got 2 different types of cells here, the principal and intercalated cells, which are going to help with this balance, this hormonal balance, as well as the reabsorption process, which we will see mainly being that sodium and potassium, and the hydrogen ions, remember, are going to be secreted back into the tubule for that pH balance. Now we're going to see, again, we've kind of talked about how the body's hydration is going to control the amount of water conservation that actually happens. And again, this is based on the body's mechanism of identifying whether it is in this dehydrated state to where it has to conserve water. And that's going to be done by primarily done by hormonal, but we're also going to see that it can uptake water or increase the uptake of water by the number of aquaporins. If you remember, the aquaporins are those transport proteins that are going to allow water to come into the cell transcellularly. If you remember, we had paracellular, which the water can come through just through those tight leaky junctions. However, we've got the aquaporins that can actually be produced, and this is actually another genetic factor that can increase. So if water uptake needs to occur, what happens is the antidiuretic hormone, the mode of action of how the antidiuretic hormone works is it actually increases the amount of these aquaporin proteins that are within the kidney cells that can increase that water uptake in the transcellular route. So here's this countercurrent multiplier that we were just talking about. This is a mechanism by which the nephron tries to concentrate solutes so it can produce water movement. Okay, so if you recall, on the thin segment of the loop of Henle, or that nephron loop, it was permeable to water. Okay, but not solutes. So what happens to the fluid, the filtrate in the thin segment of the nephron loop is it's going to become more concentrated with solutes, right? As water exits. So as water exits, we're going to see that this countercurrent multiplier means that the solute concentration is going to multiply, or it's going to become more concentrated. So we have a more abundance of water on the outside. So if you can envision the renal pyramid right here. So now the water is exiting and is in the renal pyramid or in that extracellular tissue. So we have a great abundance of extracellular fluid. So as we now are pushing the glomerular filtrate up the thick end of the nephron loop, or this ascending portion, we're going to see now that it's permeable to solutes. So as the solutes move out into the renal pelvis area, or that tissue, we're going to see that we want to concentrate the solutes on the outside. We want to do this because as we go through the collecting duct, this is where water conservation is going to occur. Well, if we have a high amount of solutes in the extracellular fluid here, the water is going to want to exit that collecting duct and move into the tissue because that's where the high concentration of solutes is. So this mechanism is called the countercurrent multiplier, and it's all based on osmotic principles. And where we want a high concentration of solutes because we want the water to flow that direction. Okay, so again, as we move down the loop of Henle, the descending loop, the thin segment is going to be permeable to water. So we're going to see water exiting the thin segment and going into the extracellular fluid. Because of this, it is now going to concentrate the solutes in the thin segment of the loop of Henle. Now, as we move up the ascending loop of Henle, or the thick segment, it is now going to be permeable to solutes, but not water. So the solutes now are going to exit into the tissue and make the concentration of solutes very, very high in the tissue. As we do this, we're going to see now the collecting duct, as it leaves the distal convoluted tubule into the collecting duct, we want the water to exit. And it's going to do this because of the amount of solutes that is found in that extracellular tissue, because of what just occurred within that loop of Henle. Okay, so again, remember, this is all based on osmotic principles. The water is going to flow where the concentration of solutes is the highest. So this is where we want to conserve water in the collecting duct just by way of osmosis. Okay, as water exits the collecting duct into the tissue, the urine is now going to be more concentrated. So this process is going to show, like we were talking about, the color of urine can change based on how much water is going to be reabsorbed. If the body is dehydrated and there's a great amount of solute concentration in the extracellular tissue because there's less water, that means that we're going to see that the urine is going to be much more of that darker amber color. However, if the body is not in that dehydrated state and there is an excess of water that can be excreted, we're going to see that there is a good amount of water that will be reabsorbed. However, it will not reabsorb all of it, and it will excrete it through the urine, and that's where the color of the urine can be a little bit more clear or light yellow.